Well, today we're here with Dr. Nazema Siddiqui from Duke University, who's an associate professor and the director of research for the Division of Urogynecology and Female Reconstructive Surgery there. She is really a blossoming leader in the field of the microbiome and understanding lower urinary tract dysfunction. And today I've asked her to come and talk with us about the role of the urinary microbiome in overactive bladder and urgency incontinence. Dr. Siddiqui, welcome so much. Thank you so much for being here today.
Well, thank you for having me. And thank you for inviting me to speak on this subject, which has sort of become a blossoming area of research interest for me as well. Here are my disclosures. Before we sort of launch into microbiology, I think it's sort of helpful to reflect on how we define overactive bladder, because that becomes relevant when we think about how microbes may interact. Multiple societies have issued guidelines that lend themselves to our current conventional definition, but it's basically this: symptoms. The definition of overactive bladder is symptoms of urinary urgency, usually accompanied by frequency and nocturia, with or without urgency urinary incontinence. And I don't think this is a surprise to people, but what we often forget is the second part that's written in the printed statement, which is in the absence of urinary tract infection or other obvious pathology. And generally, when we are trying to assess for urinary tract infection, we do this with a standard urine culture. But we are hearing more and more that the standard urine culture is, somewhat problematic, because it was really developed to optimize the assessment of gram-negative microbes. So it sort of lends itself to this question or a couple of questions really, of whether overactive bladder symptoms could result from a microbe that simply can't be detected with standard culture. So for example, if you think about gastroesophageal reflux disorder and H. pylori, before the sort of knowledge of this microbe called Helicobacter pylori, we thought GERD was a much more anatomic problem. So that's one example that could be relevant for the urinary tract. Another key question is whether or not overactive bladder symptoms could result from a dysbiosis, or sort of a shift in the ratio of normal commensal microbes to other microbes. Parallels are seen in other spheres, like bacterial vaginosis in the vagina, or small intestinal bacterial overgrowth in the GI tract. So I think these are the pressing questions in our field, or whether or not overactive bladder could actually be occurring because of these types of issues. To sort of understand why there's more and more interest in exploring this now, I do want to just speak for a moment about the Human Microbiome Project. And this was a big project funded by the National Institutes of Health, and really was sort of conceptualized many, many years ago, with samples being collected until about 2010. And during the time period of that project, non-invasive sampling was performed in many sort of cracks and crevices in our body. That really has been the impetus for a lot of research in the gut microbiome, for example, or the vaginal environment, or the skin, and many other places in the body. And to be fair, in this project, although they talk about sampling the urogenital tract, really, if you look at what was sampled, it was vaginal flora. So we have very, very little knowledge about what might be in the bladder until recent years. And unfortunately, during the Human Microbiome Project, we did not get the benefit of having the urinary tract be one of the locations of sampling. So we're a little bit behind compared to other fields, but I think there's a lot more interest now. Probably about a decade ago, when there's a lot of excitement about what was happening in the gut and other places, many people ask, well, is there a urinary microbiome? And what we can say now is pretty conclusively that yes, there is one. And this really dates back to almost a decade ago, where a group in Norway, this is led by Siddiqui et al. I wish it were me, but it is not. But this group essentially did one of the sentinel studies that helped to suggest that there are commensal microbes living in the bladder. They sampled 8 kind of standard culture negative women These were voided samples, so there's some criticism there, but essentially applied this technique called 16S rRNA sequencing. And you hear a lot about this now, but it's basically a method of identifying microbes based on their bacterial DNA sequence. So it does not rely on culture. And basically, using this technique, they found that yes, indeed, these 8 culture-negative women had bacteria living in their bladder. Now, obviously, the big criticism here is that these were voided samples. This might have been contamination from the vaginal area or distal urethra. But before I get into that criticism, I do want to just take a brief moment and explain this technique of 16S rRNA gene sequencing, because it comes up repeatedly now when we look at microbiome studies. But essentially, what you do for this technique is you take a sample wherever, from whatever body site that may be, and that sample contains a mix of human cells which have DNA, as well as bacterial cells which have DNA. And so you have to go through a process of first isolating the DNA. And one thing that becomes really important as we start to look at microbiome studies is that gram-positive DNA is a little bit harder to extract. Gram-positive bacteria have a thicker cell wall, and so sometimes, depending on the extraction methods that are being used, you could be a little bit more biased towards detecting gram-negative stuff than gram-positive. I'm going to leave that aside, but that's something that is a caveat when we're looking at the literature. But anyways, you go back to your sample. You have a mix of human DNA and bacterial DNA, and you isolate all that DNA. And then you basically use a PCR-based technique to amplify certain segments of the DNA. And in particular, we are using probes, PCR probes, that are designed for the 16S ribosomal subunit that only bacteria have, and eukaryotic cells, human cells, do not have this particular subunit in our ribosomes. And so if we can probe with PCR probes that are specific for that subunit, then first of all, we can amplify bacterial DNA and kind of pull out the bacterial segment of the DNA. And then we can further amplify segments of that ribosomal region that technically are called variable regions. And within those variable regions are areas that can sort of map back and tell you what that particular microbe may be. It kind of helps us to identify what it is, what piece of DNA we pulled out. And so essentially, you can use this technique, and as I'm kind of walking through this slide here, I've just been talking about this whole process of PCR amplification of different variable regions. And basically, you then sequence whatever you've sort of pulled out with PCR. You take your sequences, and then you have to go through some bioinformatics, or basically methods of mapping back these strings of A, C, T, and G, these strings of sequence material back to these reference databases, and it tells you what bacteria you had. The reason I went through all of this detail is because at each step of the way, there are places where you have to be careful of bias and be careful of sort of how you interpret that information. And so, many times as clinicians, when we view these studies, we see like, oh, they got these sort of bacteria out of this sample, and we interpret that as sort of that's a fixed and true thing. And we just have to be a little bit careful sometimes when we're interpreting this information, especially when it's early on. So anyways, back to sort of the story of what's happening in the bladder and the urinary tract. And coming back to that study that I mentioned, so Siddiqui et al. Applied this technique for one of the first times to urine samples and found that there's bacteria in there that we just didn't appreciate with culture. And I'm going to come back to that criticism, which is that these were voided samples, and so a lot of people thought, well, that could just be contaminant. And so this is sort of one of the sentinel studies, I think, that has really helped to propel this field by Wolf et al. But this group at Loyola in 2012 performed a really interesting study where they basically performed sampling through multiple routes in the same woman who was undergoing surgery. So basically there was a voided sample, there was a transurethral catheterized sample with a skin swab of the distal urethra, and then there was also a suprapubic aspirate with a skin swab of the suprapubic region. And basically they were able to analyze these data and tell us that, first of all, catheterization does give you more specific information for the bladder. When you have a voided sample, you have a lot of overlap with vaginal contamination, even if it's a clean void sample. And not only does catheterization a good way of detecting things, but essentially transurethral and suprapubic aspirates were quite similar, and there really were unique bacteria that were not noted in the skin swabs. So these are not thought to be contaminants, but truly bacteria that are living in the bladder. And so once we had that information, people started to really take that and run with it. The same group at Loyola has also started to pioneer another technique that's called expanded quantitative urine culture. And this has really helped us to understand some of the limitations of the standard urine culture that we use in the clinic. And this is now a pretty pervasive picture within the microbiome field, showing essentially the same urine sample that was placed on two identical plates. The one on the left was subjected to standard urine culture techniques. The one on the right was subjected to some more expanded culturing techniques, which include different levels of carbon dioxide, different media, and essentially the same urine you can see is not really sterile. The quote, no growth culture on the left, that same exact urine sample will show growth of different microbes or different bacterial colonies when it's subjected to different conditions, and you can see different morphologies, so it's not even just one. So when we start to look at this combination of 16S rRNA sequencing as well as expanded quantitative urine culture, we are basically just starting to see that there's a lot that we just didn't previously appreciate that is in the bladder, a lot of microbes that are living in there, and that these microbes may be relevant for urologic health, and particularly for overactive bladder. So that sort of brings us to the story of, how do we start to investigate the microbiome and overactive bladder. And one of the early studies to look into this was published in 2014 by Pierce et al. And in this study, they essentially looked at, these are all women who had all undergone catheterized sampling, but essentially about 60 women with urgency incontinence and 58 controls. And all of these samples underwent these two techniques that I just talked about, the sequencing technique as well as the expanded culturing technique. And in this study, the authors found that there were differences in the microbial communities between women with urgency urinary incontinence and controls. And they found differences with both techniques, although they were you can find slightly different things with each technique. So this is just a picture of what sequence data may look like, where essentially we get these bars that are color-coded for different types of bacteria. And the one on the left were the urgent continence patients. The one on the right shows the controls. And sort of the big ticket thing to take away here is that basically blue, which is lactobacillus, you see a lot more blue on the right in these controls. On the left, the urgency incontinence patients, you see more of that kind of maroon reddish color, which is Gardnerella. But it's not a pure difference here, and there's quite a bit of mixing. And these are some of the results from the same paper, particularly focusing on the cultures, the expanded culture technique that I mentioned. The importance of the cultures is that here you're growing live bacteria. Sequencing, you can assess things, they may be living or dead. And I mentioned all those steps along the pipeline where there could be some bias involved in what you recover. Here with culturing, you really are growing out live bacteria that you can test further. And again, in this study, women with urgency incontinence, there were a number of differences in the live bacteria that grew out from those urine samples compared to controls when you use expanded culture techniques. There was one interesting difference I'm going to see if I can point out here for you, which was that the one thing where the controls were higher was actually a particular type of lactobacillus, which is Lactobacillus crispatus. So that was an interesting finding. But before we get super excited about what we're seeing here, the devil is in the details. And so this is where I just want to urge people to interpret things with a little bit of caution. Just like any other research study, for microbiome studies, we also have to look at whether the populations that are being compared are comparable. And that helps us to understand what conclusions we can draw out of these studies. And in this study in particular, the table 1 was quite interesting. And if you look at it in detail, there are a number of differences between the urgent continent women and the controls. A big one was age. So the control population was significantly younger. Another one was related to estrogen status. And so there were differences in the sort of menopausal status and or who was using supplemental estrogen. And then another big one was with body mass index. And so the controls were significantly lighter in weight and body mass index than the urgency incontinence patients. And as you can imagine, age, estrogen, and body mass index can all also affect the urinary microbiome. So now we come back to the question of whether or not these factors were confounders and whether or not the associations we see with, overactive bladder and urgency incontinence, whether those associations are real or whether or not they're more influenced by these confounding factors. That is really still part of where the questions lie. One other study I do want to point out has tried to address some of that, and this is published by Lisa Karstens and her group in Portland, Oregon, and this was published in 2016. Now, Lisa Karstens is a bioinformatician, so she's not a clinician, so she gets very entrenched in the data component of things and basically worked with a team to really try to recruit 10 women with urgency incontinence and 10 tightly matched controls to try to address some of these confounding factors. They also performed catheterized sampling, but they really worked hard to match the subjects on age, BMI, and menopausal status, those three critical factors I pointed out before. Now, in this study, these pie charts, you can just kind of glance at them and say, yeah, there are some differences there. So again, we are seeing some differences in terms of the microbiota that are recovered in urgency incontinence patients and controls, even when we tightly control for some of these confounders. And so now we're getting more information and more evidence that there might be some true associations there. And furthermore, in this study, the group looked a little bit more at some of the different particular bacterial species that they recovered. This was via sequencing. And they basically found that in the women with urgency incontinence, there were 5 bacteria that were lower than controls and 9 bacteria that were actually increased compared to controls. Of those 9 that were increased, 5 of those are also somewhat associated with urinary tract infection, which again leads us back to that sort of question of whether or not there are microbes that are sort of causing irritative symptoms, things that are more on the spectrum of UTI, and whether or not those microbes are really part of the etiology of overactive bladder in a subset of patients. So, really, I think that we have these key questions that are existing in our field. Could overactive bladder symptoms result from a microbe that can't be detected with standard culture? I think I've shown some evidence that yes, that may be the case. And then also, could overactive bladder symptoms result from a dysbiosis or a shift in some of the commensal microbes? Well, that is quite plausible too. So really, to answer these questions, we need to start to understand what are some of these differences that we're detecting between different populations really mean? How might microbes actually interact with their host to produce irritative symptoms? So what are some of the mechanisms behind why certain bacteria may produce irritative symptoms compared to others? And those sorts of questions and answers may help us to better understand if there's really a microbial etiology or a microbiome etiology to a subset of women and men who have overactive bladder. So really just to summarize sort of what I hope you can take home from this interview today, I think that we have some pretty conclusive evidence that a urinary microbiome exists. We are starting to see multiple association studies that show some differences in microbes in patients who have overactive bladder versus controls. We have to be very careful about how we interpret that literature because we have to think about things like age, sex, menopausal status, body mass index as potentially confounding factors that we have to think very critically about when we're interpreting the data. However, we may start to see differences in microbes in these conditions, and we may start to see differences at the species level, and those may really warrant some further follow-up in the future.
So I have one quick question. This was a wonderful presentation, and I wanted to just kind of get your thoughts about how we start to get at the relationship between overactive bladder symptoms and these urinary microbes, because as you sort of pointed out, a lot of this stuff is very correlative. And how do we move from correlative to actually beginning to start making some progress on understanding how this might work or how we might use this for therapy?
Oh, that is a really great question. In my mind, I think there's a couple of things that need to happen. First of all, we have correlations and associations, but really designing some longitudinal studies that better assess the microbiome over multiple periods of time, and particularly as as patients are transitioning through different phases of symptoms, that would help us a little bit more, or a lot more. So, I do think that some form of longitudinal sampling, longitudinal studies are really going to be important. Um, Within the microbiome field, a lot of people have taken to using animal models or these organoid models, basically ways of testing microbes and what they do to cells. One particular area of interest are using what we call gnotobiotic animals, which are um, usually rodents, but animals that are raised in a germ-free facility, and so essentially, don't have the native microbiome. And in those instances, you can introduce a human profile, you can introduce a dysbiotic profile, you can introduce all sorts of different things and test associations. And I think those kinds of studies are going to be really helpful too, because just like many areas of biology, I don't think we're going to find that it's just one microbe or one particular thing that is altered. I think it's probably a little bit of a complex situation where you may have shifting of ratios, or you have, a little bit less of the good stuff and more of the bad stuff. And to really kind of test those sorts of profiles, you probably do need to move to some sort of model system where you could understand that a little bit better.
Yeah,
So
and control those factors a little bit better.
exactly. Yeah, I
Hmm.
think it's going to be a mix of some longitudinal studies in humans, as well as going into some of these model systems where you can really experimentally design what microbes are present and not present, and then, kind of putting those pieces of information together.
That's amazing. Do you now to wax a little poetic here, do you foresee a future in which we will have microbiome targeted therapies for overactive bladder? Do you think that's something we can dream about?
I hope so. I think that we are probably getting closer to that in the recurrent urinary tract infection world. I suspect that will come first. But, I think many of us who treat these patients, and I tend to treat women, so I talk about this in terms of women, but, many of us who treat these patients really see this sort of subset that seems more infectious, where there's this, and we know that there's an overlap of a certain subset of people with overactive bladder seem more neurologic, but then there's a
Hmm.
certain subset that seem a little bit more like infectious. They also have recurrent UTIs. They say that their symptoms get worse when they have a UTI.
Yeah.
Sometimes when they're on their prophylactic antibiotic, they say all their OAB gets better.
Hmm.
So that particular profile, I have a little bit of hope that if we can hone in on what's happening with that little subset of overactive bladder, that may be a place where we could intervene with a therapeutic.
That's wonderful. Thank you so much for your time. I really appreciate you spending the time to discuss this with us today.
Absolutely. Thanks for having me.